Imagine a world where cells couldn't divide, DNA couldn't replicate, and life itself would come to a screeching halt. The culprit? A missing enzyme. One so fundamental to life that without it, evolution itself would have stalled. That enzyme is ribonucleotide reductase, but we're going to refer to it as RNR for short. Today we're diving into the science behind this molecular workhouse and its profound impact on medicine and biotechnology. RNR is the enzyme responsible for converting ribonucleoside diphosphates, the RNA building blocks, into deoxyribonucleoside diphosphates, the DNA building blocks. It does this by reducing the ribonucleoside diphosphates, removing their 2' hydroxyl group. This process is essential for DNA replication and repair, meaning no RNR, no DNA synthesis, no cell division. RNR is a complex multi-subunit enzyme that operates through radical-based chemistry. It has 2 main subunits. The R1 subunit, which is made of 2 alpha subunits, which each contain the catalytic site with cysteine residues and regulatory domains controlling enzyme activity. The R2 subunit is made of 2 beta subunits with an iron cluster that generates a free radical to form the critical tyrosine radical site. The reaction starts when the free radical is formed and passed to the tyrosine radical site. From there, it is passed onto one of the R1 cysteine residues in the R1 active site. Next, the radical at the 3' carbon of the nucleotide helps remove a water molecule from the 2' carbon, preparing the nucleotide for reduction. Another cysteine donates a hydrogen atom to the nucleotide at the 2' carbon, forming a disulfide bond between 2 cysteine residues in the enzyme. The radical is then transferred back to the first cysteine. The new deoxyribonucleoside diphosphate is released, but leaves RNR in an inactive oxidized state. To become functional again, it is reduced by thioredoxin reductase using NADPH, allowing the cycle to repeat. This reaction requires tight regulation, since an imbalance in deoxyribonucleotide pools can lead to genomic instability and disease. RNR activity is controlled through 2 allosteric sites. One is the activity determining site, which is like an on and off switch. ATP binds when ribonucleotide levels are high, activating RNR, allowing deoxyribonucleotide production. DATP binds when there are enough deoxyribonucleotides, inhibiting RNR and preventing excess production. Then there's the specificity determining site, which controls which nucleotides are reduced. ATP binding promotes the reduction of CDP and UDP. DTTP binding shifts RNR activity to reduce GTP, and DGTP binding directs RNR to reduce ADP. Once all these steps are complete, all 4 deoxyribonucleotides are produced in balanced amounts for DNA synthesis. So why should we care? Beyond its fundamental role in biology, RNR is a critical target in medicine, especially in cancer research. Cancer cells are addicted to DNA replication. Since they divide uncontrollably, they require a constant supply of deoxyribonucleotides. This makes RNR a prime target for chemotherapy. Drugs like hydroxyurea and gemcitabine inhibit RNR, starving cancer cells of the DNA precursors they desperately need. Without these building blocks, cancer cell growth is halted. But there's a challenge. Cancer cells can develop resistance to RNR inhibitors. Researchers are now investigating new drug strategies, including combination therapies and novel inhibitors, to outsmart cancer's adaptability. Understanding RNR is opening doors to more effective, less toxic treatment. Thank you, and next time you think about DNA, remember the tiny enzyme making it all possible.